One of the topics that are commonly taught in a biochemistry course are nucleic acids. Now you might be wondering, what exactly are nucleic acids? And there are two forms of nucleic acids. I'm sure you heard of them, DNA and RNA. DNA stands for deoxyribonucleic acid. RNA is simply ribonucleic acid. But both of these are different types of nucleic acids. As you can see, DNA is a double-stranded nucleic acid. It forms an alpha helix, whereas RNA is a single-stranded nucleic acid. So that's one difference between the two. There are other differences. Now, DNA is mostly located in the nucleus, particularly in eukaryotes, whereas RNA, you could find it outside of the nucleus. And DNA basically, its function is to store genetic information. It serves as the library of the cell, whereas RNA, it can be used to transfer genetic information from one part of the cell to another, and it can also be used to synthesize protein. There's different types of RNA. You have ribosomal RNA, transfer RNA, and messenger RNA. So if you see tRNA, that's transfer RNA. If you see mRNA, that's messenger RNA. And then rRNA is ribosomal RNA. DNA and RNA are both polymers. Now you might be wondering, what exactly is a polymer? A polymer is basically a long molecule made up of tiny units called monomers. And the monomers that make up DNA and RNA are known as nucleotides. Now there's different types of nucleotides, but they all have three basic parts to it. So each nucleotide has a pentose sugar, a 5-carbon sugar, a nitrogenous base, and a phosphate group. So P stands for phosphate, S is for sugar, B is for base. Now in DNA, the type of sugar that we have is a 5-carbon sugar known as deoxyribose. Now I'm going to compare it to RNA so you can see the difference. RNA has a ribose sugar instead of a deoxyribose sugar. It still has a nitrogenous base and it has a phosphate group. But the difference is an extra hydroxyl group. Now let's go ahead and number the carbons on the ribose sugar. This is carbon 1, that's where the nitrogenous base is attached to. Carbon 2, 3, 4, and carbon 5 is outside of the pentose ring. And that's where the phosphate group is attached to. So in DNA, notice that we don't have a hydroxyl group on carbon 2. And so that is why it's called deoxyribose. It's a deoxyribose sugar because it's lacking in oxygen. Whereas in RNA, you have a ribose sugar. That's why RNA is called ribonucleic acid, but DNA is called deoxyribonucleic acid. But besides that, they both have a phosphate group and a nitrogenous base. Now let's focus on the nitrogenous bases found in DNA and in RNA. So DNA contains the bases adenine, guanine, cytosine, and thymine. In RNA, the bases are adenine, guanine, cytosine, but instead of thymine, it's uracil. And so that's another difference between RNA and DNA. It's a typical test question, so make sure you're aware of that difference. So uracil is found in RNA, and thymine is found in DNA. Now the nitrogenous bases that we've been talking about can be divided into these two categories, purines and pyrimidines. Now the purines contain 2 rings. One of the ring is basically a 6-membered ring, and the other one is a 5-membered ring. Pyrimidines, on the other hand, contain only one ring, and it's a six-membered ring. Now, the purines that you need to be aware of are adenine and guanine. The pyrimidines include cytosine, thymine, and uracil. Now let's talk about drawing these structures and also how to number them. For some of you, you may need to be able to draw these structures on your tests. And for those of you who don't need to memorize it, you can fast forward this section, or you can sit back, relax, grab a bag of popcorn, and just enjoy the show. So let's start with the purines. On the left side, I'm going to draw adenine, and on the right side, I'm going to draw guanine. Now, in the first ring, they both contain 2 nitrogen atoms and 4 carbon atoms. In the second ring, they also contain 2 nitrogen atoms. So both adenine and guanine has that same structure. So if you start with the base structure, it can help you to remember how to draw these 2 purines. Now both adenine and guanine, they have a double bond in the middle, and they have another double bond in this position and here as well. Now let's talk about where they're different. By the way, there's a hydrogen here, so that's a NH. If you don't see a hydrogen, that means that there's a lone pair on the nitrogen. Now adenine has an NH2 group on this position right here. So that's where it's different relative to guanine. Guanine has an NH2 group towards the right. And at the same time, it has a carbonyl group in this location. So if you remember that, it can help you to draw these 2 structures. So that's the difference between adenine and guanine. Now let's talk about numbering the rings. So this nitrogen represents number 1, and then you need to count it in a counterclockwise direction. So this is 2, 3, 4, 5, 6. Then you move on to the 5-member ring. This is 7, and we're going to count it in a clockwise direction. So this is 8, and this is 9. Now, what you need to know with purines is that when you attach them to a ribose, they will be connected. Let me draw a ribose ring. I'm just going to draw a box to represent a ribose, just to keep it simple. But what I want you to take from this is that the ribose ring is attached to the nitrogenous base at position 9 when dealing with purines. For pyrimidines, it's different. But for purines, the ribose is attached to the N9 position. So it's the 9th position on a nitrogen atom on a purine ring. So you may need to know that for a test. Maybe not. It depends on if your teacher's going to quiz you on that fact. I don't know. Now let's move on to the pyrimidines. So there's 3 structures that we need to draw. Now let's start with the first one. So once again, we're going to have a 6-membered ring with 2 nitrogen atoms and 4 carbon atoms, just like we had in the case of the purines. We only have a 6-membered ring here, but we're not going to have the 5-membered ring for the pyrimidines. But the structure of the 6-membered ring is very similar. So on the left, I'm going to draw, let's start with thymine. And then in the middle, I'm going to draw uracil, and then cytosine. So all three of these nitrogenous bases, they have the same general structure. The six-membered ring, as you can see, it's very similar. Now let's talk about the way we're going to count it. So this is going to be number 1, and we're going to count it in a clockwise direction. So this is 2, 3, 4, 5, and 6. Now the ribose will be attached to the nitrogenous base at the N1 position. So make sure you keep that in mind if you're taking notes. Now I'm going to get rid of the numbers, but you can add it there in your notes. So this is going to be a hydrogen in the case of thymine, and we're going to have a double bond between positions 5 and 6. And the same is true for the rest of them. Now for thymine, we have a carbonyl group at position 4 and another carbonyl group at carbon 2. And here we have a hydrogen. So that's thymine. Now uracil looks very similar to thymine with one key difference. Thymine has a methyl group, but uracil does not. And so that's the difference between thymine and uracil. Uracil has a hydrogen. If you don't see it, it's an invisible hydrogen, but it's there. Now, in the case of cytosine, we have a carbonyl group on carbon 2, just like thymine and uracil. So they're similar in that respect. However, we do have something different in cytosine, and that is we have an NH2 group on carbon 4. And so that's how cytosine differs from thymine and uracil. It has this NH2 group in this position. In addition to that, it also has a double bond between positions 3 and 4. So those are the 3 pyrimidines that you may need to know. And just remember, uracil is found only in RNA, thymine is found only in DNA, and cytosine is found in both DNA and RNA. Now here's a question for you. What is the difference between a nucleotide and a nucleoside? What would you say? Perhaps you heard of the word nucleoside. What really is a nucleoside? Well, let's go back to nucleotides. We know that a nucleotide has 3 parts. As mentioned before, it has a ribose or a deoxyribose sugar. It has a nitrogenous base, which could be a pyrimidine with 1 ring or a purine with 2 rings, and it also has a phosphate group. Now, in the case of a nucleoside, it doesn't have 3 parts. A nucleoside has 2 parts. It has the 5-carbon sugar, and it has the nitrogenous base. It does not have the phosphate group. So this is a nucleoside. It's the sugar and the nitrogenous base. But once you add the phosphate group to a nucleoside, then it becomes a nucleotide. So make sure you understand the difference and also the naming. So let's say this is cytosine, which is one of the 3 pyrimidines we talked about. Now, by itself, cytosine is a nitrogenous base. Now let's focus on the nomenclature. So this is cytosine. When you add the ribose sugar to it, it becomes a nucleoside, and it's called cytidine in the case of RNA, but in the case of DNA, it's deoxycytidine. And it really depends on the presence of the OH group on carbon 2. So this is 1, 2, 3, 4, 5. There's always going to be an OH group on carbon 3. But if we don't have an OH group on carbon 2, it's called deoxycytidine. If we do have it, then it's called cytidine. Now let's think about this. If the nitrogenous base is called cytosine, and if the nucleoside is called cytidine, what is the name of the nucleotide that contains the cytosine nitrogenous base? So it's going to sound weird, but it's called citidylate. Chances are you probably don't need to know that for your tests, but for those of you who may need to know it, that's what the nucleotide is called. So when you hear the word cytosine, it's not referring to this entire nucleotide, but rather just the base that's in the nucleotide. But if you hear citidylate, that's the entire nucleotide. So here is a summary that can help you with the nomenclature of nucleosides and nucleotides. On the left, we have the nitrogenous base adenine. Once we add a ribose sugar to it, it becomes adenosine. And then if we add the phosphate group, it becomes adenylate. Looking at guanine, the situation is similar. As a nucleoside, it's guanosine, and as a nucleotide with the phosphate group, guanylate. Now the nitrogenous base thymine, once we add a ribose sugar to it, it becomes thymidine, or thymidine, if that's how you say it. But since thymine is found in DNA, chances are it's going to be added to a deoxyribose sugar, and so it becomes deoxythymidine. And instead of thymidylate, it becomes deoxythymidylate in DNA. My pronunciation of these names may not be the best, so don't quote me on that. You can look it up yourself, but these are the names that corresponds to the nitrogenous base, the nucleosides, and the nucleotides. Now let's talk about naming nucleosides. How would you name this particular nucleoside? So the first thing we need to do is identify the type of nitrogenous base that we have. Now based on the structures that we drew earlier, what kind of base do we have? Well, first, is it a purine or is it a pyrimidine? Now, if you recall, pyrimidines are nitrogenous bases that have only one ring. So because this is a two-ring nitrogenous base, we have a purine. Now, there's two purines you need to be familiar with, and that's adenine and guanine. So which one is this? Is this adenine or guanine? So this particular nitrogenous base is called guanine. Now, what does it become once we add a sugar to it? Guanine plus the ribose sugar becomes the nucleoside guanosine. And so that's what we have here. This is position 1, 2, 3, 4, 5, 6, 7, 8, 9. So as we can see, the purine is attached to the ribose at the 9th position on the nitrogen atom. Now, what happens to the name if we put, let's say, a methyl group on carbon 8? How do we name this particular nucleoside? Well, this becomes 8-methyl guanosine. Now, what if we remove a hydroxyl group? Let's say if we replace this with a hydrogen, as in the case of DNA. What does it become now? So now we have a deoxyribose, and so this is going to become deoxyguanosine. This is carbon 1 on the ribose sugar. This is 2, 3, 4, and this is carbon 5. So now, the way we're going to name it, I'm running out of space here, so let's see if I can fit it in. It's going to be 8-methyl dash 2-deoxyguanosine because we don't have the oxygen, or the hydroxyl group rather, on carbon 2 of the ribose sugar. So now it's deoxyguanosine. And so that's a simple example of how you can name a nucleoside with a purine ring. Let's try another example. So how can we name this particular nucleoside? So first, let's start with the nitrogenous base. Now, we have a one-ring nitrogenous base, so that means it's a pyrimidine, and we have three options. It's either cytosine, thymine, or uracil. Now, looking at the NH2 at the top, only one of those three pyrimidines have the NH2 on the top. And if you remember, this is the nitrogenous base, cytosine. Now, combined with the ribose sugar, it becomes a nucleoside called cytidine. And so that's the name of this particular nucleoside. Now, how will the name change if we add a methyl group to that carbon? So we need to number it. This is 1, 2, 3, 4, 5, 6. So this becomes 6-methyl cytidine. And so that's how we can name it. And also, remember that the ribose is attached to the pyrimidine ring at the N1 position. For the purine, it was the N9 position. So just keep that in mind. Now let's shift our focus to naming nucleotides. So we're going to have our ribose sugar, our nitrogenous base, and a phosphate group. So how can we name this particular nucleotide? So the nitrogenous base that we have, this is G, and so that's guanine. When we combine it with the sugar, it becomes a nucleoside called guanosine. But now, how do we name it once we have the phosphate group? So it becomes guanylate when the phosphate group is at position 5. Another way in which we can name this particular nucleotide is we can start with the name of the nucleoside, guanosine, and then specify the location of the phosphate. So we could say guanosine-5-monophosphate because we have one phosphate attached to the ribose sugar. And so that's how we can name this particular nucleotide. Now let's try another example. So this time, the nitrogenous base that we're going to use is adenine. And the phosphate group is going to be placed in a different position. So you can also see phosphate represented this way. So how can we name this particular nucleotide? So given the base adenine, once we combine it with the sugar, it becomes the nucleoside adenosine. Now the phosphate group is located on carbon 3 of the ribose ring. So to put it together, we're going to start with the name of the nucleoside, adenosine, and then specify the location of the phosphate group. So adenosine-3-monophosphate. And so that's how we can name that particular nucleotide. Now let's try another example. So we're going to use the same nitrogenous base, adenosine. But this time, we're going to have multiple phosphate groups. So let's say if we have 3 of them, how do we name it now? So we're going to start by naming the nucleoside. So if we combine the ribose sugar and the nitrogenous base, that is called adenosine. Now we have 3 phosphate groups, and so it's going to be called triphosphate, tri for 3, and it's located on carbon 5. So it's adenosine dash 5 dash triphosphate. Now this particular nucleotide has a common name, and so sometimes the 5 is just ignored because it's a very common molecule, and so it's simply referred to as adenosine triphosphate. And perhaps you heard of it as this molecule, ATP. So that's adenosine triphosphate. Now there's some other ones. For instance, if you hear the word ADP, it stands for adenosine diphosphate. So instead of having just 1 phosphate group or 3, now you have 2 phosphate groups. So that's adenosine diphosphate. And if you hear the abbreviation AMP, this is adenosine monophosphate. So there's just 1 phosphate group instead of 2 or 3. So what I'm going to do at this point is draw a representation of a DNA strand. So here we have our sugar attached to a phosphate group, and then this is going to be attached to a nitrogenous base. So let's put cytosine in it, and over here it's going to be attached to another phosphate group, and then that's going to be attached to another ribose sugar, actually deoxyribose since we're dealing with DNA. And so this is going to be adenine this time, and then we're going to have another phosphate group. And let's put thymine in this box. Now at this point, go ahead and draw the complementary strand on the right side, using the left side as a starting point. So first, let's draw the nitrogenous bases that will pair up with C, A, and T. And then attached to each nitrogenous base, we have the sugar, but notice the direction of the sugar, how it's like pointed up. For the complementary strand, it's going to be pointed down. So we're going to draw it this way. And then we're going to have a phosphate group attached to it. And here we have another phosphate group, and then another sugar unit. We're going to talk about the connectivity of the phosphate group shortly. I just want to complete this first. So, what bases will go in these boxes? Now, what you need to know is that C, cytosine, will always pair up with G, guanine. So we need to put G inside this box. Adenine, A, will always pair up with thymine in DNA. And T will pair up with A. Now, the next thing you need to put are the hydrogen bonds located between these base pairs. And so it's the hydrogen bonds that keep the 2 strands in DNA attached to each other. So there's 3 hydrogen bonds connecting C and G. Now between A and T, there are 2 hydrogen bonds holding them together. Now, if you recall from chemistry, hydrogen bonds exist whenever hydrogen is attached to elements such as oxygen, nitrogen, or fluorine. And an H bond is basically an intermolecular force that exists between separate molecules. Now, for those of you who may want to review on that, if you do a YouTube search, type in intermolecular forces organic chemistry tutor, you should see a video that will give you a review on intermolecular forces and dipole interactions and things like that. Now, on the left side and also on the right side, this is known as the sugar phosphate backbone. And it makes sense. You have your phosphate groups on the left attached to the sugar units. Now, on the sugar, this is carbon 1, 2, 3, 4, and 5. So notice that the phosphate group is attached to carbon 3 and carbon 5 of the sugar units. And so this is called a 3,5 phosphodiester linkage. And it's a covalent linkage. A covalent bond is whenever two atoms are connected to each other by means of sharing electrons. So whenever two elements or two atoms come together by sharing electrons, they form a covalent bond. Now the next thing I want to mention is that these two strands, these complementary strands, they are anti-parallel to each other. So the strand on the left, it runs in the 5 to 3 direction. Now the strand on the right, it goes in the opposite direction. So if we focus on the top sugar unit, this is carbon 1, 2, 3, 4, 5. So notice that it's going in the 5 to 3 direction, but in the other way. So thus, these two strands are anti-parallel. They run in opposite directions. And so these are some basic things that you need to know if you have a test coming up. You never know which one of these facts you might be tested on, so just make sure you know that stuff. Now, I have one more question for you. Consider this particular strand of DNA. Write the sequence for the complementary strand. Feel free to pause the video and try it. Complementary to 5 is 3. And you need to know that A pairs up with T and C pairs up with G. So here we have A. We're going to pair it up with thymine. And here we have T. And let's pair it up with adenine. And C, we're going to pair it up with guanine. And A is going to go with T. G, we're going to pair it up with C. And so forth. So this is the complementary strand. And that's how you can write the sequence, by knowing this piece of information. Well, that's basically it for this video. Hopefully you found it to be helpful, and if you want more videos like this, feel free to subscribe, and don't forget to click the notification bell so you can receive updates on any videos I'm going to post in the future. Thanks again for watching.